Hello, and thank you for joining us today for BioTek's webinar event. My name is Lindsay, and I will be the moderator for today's webinar. The audio for today's webinar will be coming from your computer speakers, so please be sure they are turned on and the volume is up. To ask a question, please use the chat feature. It is located in the bottom left-hand corner of your screen. You can send in your questions at any time. Our speaker will address the questions during the Q&A session at the conclusion of the event. If we do not get to your question, we will do our best to respond by email. Today's webinar will be presented by Brad Larson, Principal Scientist at BioTek Instruments. We'd like to thank him for joining us today. With that, I'll turn things over to Brad to get us started.
Thank you, Lindsay, and good afternoon, everyone. First of all, on behalf of BioTek and our collaborative companies in this project, Corning Life Sciences, SysBio, and Enzium, I'd like to thank you for attending the webinar today. The goal is to introduce you to combined solutions that enable the performance of phenotypic tumor invasion assays as well as mechanism of action analyses that provide high throughput, accurate, real-time results through the incorporation of cellular imaging and kinetic microplate reader detection. Metastasis, or the spread of cancer cells from the site of the original tumor to secondary locations within the body, is linked to approximately 90% of cancer deaths. Therefore, not surprisingly, it is one of the most highly studied processes in all of cancer research. However, the development of therapies designed to forestall the metastatic activity of tumors has been met with multiple challenges. These include the incorporation of inappropriate tumor cell models, such as but not limited to single cancer cell lines plated on the bottom of a microplate in a two-dimensional or 2D format, mechanism of action determinations which do not include relevant analysis procedures, and inaccurate tracking and quantification of microscopic tumor invasion. Therefore, new cell models and methods of analysis are required to reduce the number of failures once lead molecules reach clinical trial. Tumors in vivo exist as a three-dimensional or 3D mass of multiple cell types, including cancer and stromal cells. Therefore, incorporating a 3D spheroid-type cellular structure that includes co-cultured cell types forming a tumoroid can provide a more predictive model than the use of individual cancer cells cultured in a traditional 2D format. Second hurdle, as I mentioned previously, is accurate mechanism of action determination. Possessing accurate knowledge of how invasion is interrupted provides a more complete picture before proceeding to clinical testing. Real-time analysis of matrix metalloproteinases activity during the 3D invasion process following inhibitor addition, in addition to kinetic examination of inhibitor receptor resonance times in comparison to ligands whose binding is known to contribute to the metastatic process, also yield mechanism of action determinations with a higher degree of accuracy. These, coupled with sensitive detection and quantification of binding events, changes in tumoroid structures and protease activity, allow for the generation of in vitro results that have a much closer correlation to what is then generated during in vivo clinical testing. I'll start the introduction to these combined solutions by first telling you about the instrumentation used to carry out the kinetic determination of receptor ligand resonance times and imaging of the 3D tumor invasion process, followed by the microplate technology from Corning Life Sciences incorporated for tumoroid formation and performance of the 3D tumor invasion assay procedure. Then we'll look at how the Cytation 5 and spheroid microplates were used together in combination with BioTek's Gen5 data analysis software to easily and accurately observe and quantify changes to the tumoroid structure following chemoattractant and inhibitor additions. Then finally, I'll explain how we were able to perform two different types of mechanism of action evaluation. The first, looking at matrix metalloproteinase activity during tumor invasion using the INSENSE technology from Enzium, and the second to determine receptor resonance times of a known ligand and potential inhibitory compounds using HTRF TAG light technology from SysBio. The Synergy Neo was used to perform the kinetic microplate reads necessary to calculate receptor resonance times. The Neo is a multimode reader capable of being used either for screening or in core laboratories. The instrument contains dual matched PMTs above the plate stage for simultaneous capture of two separate emission signals, such as the signal from the donor and acceptor molecules used in HGRF assays, as well as a single PMT below the plate stage for use with live cell assays. NEO can also be equipped with dual injectors, which allows for direct dispensing into microplate wells while the plate is already in the reading chamber. In the assay performed here, we injected cells into wells already containing ligand or liganded compound to allow the binding process to then begin. The combination of these two features created an ideal solution for rapidly capturing the changes in fluorescent signal from both molecules as receptor binding occurred. The Cytation 5 was then used to perform the cellular imaging during all tumor invasion assays which were run. With its digital wide field imaging capabilities and multiple imaging modes, including fluorescence, bright field, color bright field, and phase contrast, the instrument can be used to capture images of a wide variety of samples, including cells, plant or insect sections, zebrafish larvae, and H&E stained tissues. The Cytation 5 is also a multimode instrument and contains all the capabilities of a microplate reader. The filter-based detection system seen in the upper right-hand corner of the instrument cutout in blue is a direct optical system which gives the greatest sensitivity for use with weakly fluorescent probes. The monochromator-based detection system seen in the upper left-hand corner in brown gives the greatest flexibility. With the incorporation of variable bandpass selection, a wide range of wavelengths can be incorporated into the read step to ensure that the highest quality results are then achieved. The digital microscopy system seen in yellow is then housed below the microplate stage. Here you see a more detailed view of the inverted fluorescence microscopy system. Up to 6 separate objectives can be placed into the instrument at a time, ranging from 1.25 to 60x. Separate phase objectives can also be included should phase contrast imaging be necessary. Imaging filter cubes are then used to provide excitation and ensure correct wavelength selection. Light is emitted from the colored LEDs, passes through an excitation filter, and is then piped up through the objective to the sample. The emission signal then passes back through the objective, is directed through the emission filter to the camera, which contains a Sony 16-bit CCD chip. The LED excitation source and filters are contained in separate parts of the filter cube, which allows the parts to be interchangeable, giving increased flexibility to image a wide variety of fluorescent probes. Gen 5 software is then used to program a variety of imaging steps, such as single image capture, image montages, or Z-stacking, such as what we're seeing here. In this step, multiple images can automatically be captured at different Z heights through the sample. A variety of algorithms can then be used to create a projected image that contains the most in-focus portions of each individual image captured. And as you'll see later in the webinar, this can help to ensure that the most accurate results are generated from the experimental procedure. Moving on from the instrumentation, as mentioned previously, Corning's spheroid microplates were then incorporated into the 3D tumor invasion procedure. The plates utilize Corning's ultra-low attachment surface to prevent cell adhesion to the microplate. In addition, the unique round well bottom design enables the positioning and growth of a single spheroid per well that are uniform and reproducible in size. The microplates are available in standard SBS footprint dimensions for 96 and 384 well formats, and the clear bottom also allows for easy visualization and imaging. The extended black sidewalls also help to reduce crosstalk and background noise for fluorescent and luminescent-based assays. Finally, due to the microplate design, no transfer step is necessary between the completion of spheroid formation and the beginning of the assay procedure, which can make the entire process simpler to perform. The ultra-low attachment surface used in the plates is a hydrophilic, neutrally charged hydrogel coating that inhibits specific and non-specific immobilization, which forces the cells into a suspended state, enabling 3D spheroid formation. The coating is stable, non-cytotoxic, biologically inert, and non-degradable. Shown here is an example of how cell morphology and aggregation varies between different plate coatings and geometries. On the far left are HT29 cells cultured for 72 hours in a 96-well tissue culture treated microplates. And as you can see, this yields the typical adherent two-dimensional monolayer of cells that we're accustomed to seeing. Upon culture in a flat bottom, ultra-low attachment 96-well microplate, these cells then begin to form multiple cell aggregates. However, due to the flat bottom configuration, the spheroids are non-uniform in size, which can lead to variability in the results seen from treatment of each spheroid as well as within replicate wells. Then finally, on the right, when the cells are cultured in the 96-well format of the spheroid microplates, a single nicely rounded spheroid is formed in each well. This leads to greater uniformity in the data from replicate wells and across the entire plate when used for screening purposes. This slide and then the video that I'm going to show next demonstrates how spheroid formation can easily be tracked in the wells of the spheroid microplate using the Citation 5. This is an example where 5,000 HCT116 cells were added to one of the wells of the spheroid microplate that I just mentioned, and then imaging was performed once per hour over 24 hours using the Citation 5 with a 4x objective. And here I'll start the video. So again, you can see over time how the cells easily aggregate into a single spheroid or tumoroid type of a configuration. Then following the completion of aggregation, these can then be used for performing whatever assay that you see fit. We then went on to create the following procedure to perform the entire multi-day 3D tumor invasion experimental process. MDA MV231 breast cancer cells expressing GFP and human dermal fibroblasts expressing RFP were combined in a 1-to-1 ratio and dispensed into the wells of the spheroid microplates to create a final concentration of 5,000 total cells per well. Following tumoroid formation, a portion of the media was removed and replaced with media containing chemoattractant or chemoattractant and inhibitor. Diluted matrigel matrix, again containing either chemoattractant or chemoattractant and inhibitor, was then added on top of the aqueous layer as an overlay. Then following centrifugation to reposition the tumoroid in the center of the well, fluorescence and bright field imaging was then performed for the initial time point and subsequently repeated on a daily basis throughout the entire incubation period. Then to make sure that everyone has a firm grasp of what's really going on in the well during this process, I'll go over the procedure one more time in a more pictorial manner. So here we see again the empty well of the spheroid microplate. Cells in media are added to the well and aggregate into the tumoroid structure that you see in the bottom right corner of the screen. You can also see the distribution of the GFP expressing breast cancer cells and RFP expressing fibroblasts within the total tumoroid structure. After aggregation is complete, 70% of the spent media is then removed and replaced with an equal volume of media containing chemoattractant. Diluted matrigel is then added as an overlay, again containing chemoattractant. Centrifugation in a cooled centrifuge then repositions the tumoroid in the center of the well and initiates the mixing of the two layers. And again, the same procedure is followed in the case of running an inhibitor experiment, with the difference being that an inhibitor is also added along with the chemoattractant to the aqueous and matrigel matrix layers. Image-based tracking of tumor invasion is then performed for either type of experiment on a daily basis. Here in this slide, we see an example of what the tumor invasion process looks like over a 5-day incubation period. Imaging was conducted each day using a 4x objective of a tumoroid in a well containing the chemoattractant CXCL12 and no inhibitor. Each picture is actually an overlay of individual images that were captured using bright field, GFP, and RFP imaging channels. The bright field image shows the state of the entire structure, including cells and invatopodia, while the GFP and RFP images display the extent of invasion of either the MDA-MB231 breast cancer cells or fibroblasts, respectively. Then through the incorporation of BioTek's Gen5 software, which is used to not only control the imager, but also contains a data analysis package, the extent of invasion can then be determined using internal algorithms in addition to cellular analysis parameters such as bright field, signal threshold, and minimum and maximum object size. A detailed mask is automatically drawn around the object that meets the criteria. Here you can see with the arrows in gold or yellow the mask that was drawn around the tumoroid initially following chemoattractant addition and then following the 5-day incubation process. So again, as I just mentioned, you're looking at the bright field images, and you see with the use of the object mask that the software draws around the object, meeting the criteria of the different parameters that were selected, and then from the data generated, we can then perform multiple different types of analysis. The first uses the area within each object mask, and the second uses the total bright field signal within the object. And those are shown in the two graphs to the right of the images that you see here and here. And what I've done for each graph is to plot either the area or bright field signal within the object on each successive day, as you can see by the bars in red. And then the values are plotted using the left y-axis. And then I've also computed a ratio of the values from days 1 through 5 compared to the value from day 0 to normalize the results. Those are seen in green and are plotted on the right y-axis. And then with either type of evaluation, it's obvious that the tumoroid is invading at a high level through the matrix over the total incubation period. This type of analysis is useful if the cells you're using do not express any fluorescent proteins, or if there's a desire to not include any type of fluorescent probe, such as a long-term tracker dye. And in this way, this then is a truly label-free type of analysis. However, if the cells you are using do express fluorescent protein or a fluorescent probe has been added here, again, as I mentioned, the breast cancer cells are GFP expressing, the signal from the fluorescent probe can also be used to track invasion. Here we're seeing, again, in images from the signal of the GFP expressing breast cancer cells, which are captured using the GFP imaging filter cube. And the advantage here is that you can examine the extent of invasion specifically for this particular cell type. The same types of cellular analysis performed using the bright field signal to draw the object mask can then also be performed using the GFP signal. Again, we see images captured on day 0 and day 5 with the object mask automatically drawn. The graph on the top right shows the area within all the masks drawn around the images captured on each subsequent day of incubation, as well as the ratio of areas compared to day 0. Then if we look at the graph on the bottom right, we can see the comparison of the area ratios generated using either the bright field or the GFP mask, which again allows you to see how the migration of the breast cancer cells compares to the total invasion of the tumoroid structure. While using single Z-plane images is an easy and rapid way to perform the analysis of tumor invasion, it may not always provide the most accurate results due to the fact that invasion is taking place not only in the X and Y axes, but also in the Z axis. To overcome this complication, a Z-stacking imaging step can be performed in Gen5. Here, multiple images are captured at successive Z-planes to ensure that different areas of the structure are in focus with at least one image. algorithms can then be used to create a Z-projected final image that contains the most in-focus portions of each pixel included in the image. And here in this next video clip, what you're seeing are images, or different bright field images that were captured focusing on a number of different Z-planes. And obviously it's plain to see that not all of the parts of the entire structure are in focus in any individual image that was captured. Then, if we look at the final projected image, it's easy to see how a much more in focus image is created of the tumoroid structure, and this can then yield the most accurate results when performing the cellular analysis to measure invasion. And finally, the extent of migration for each cell type co-cultured into the tumoroid structure can also be examined. Here again, we see Z-projected overlaid images of our tumoroid on day 0, seen here, and then on day 5. And then with the use of the line profile tool, we can draw a line from one side of the image to the other through the tumoroid structure. And the bright field or fluorescent signal captured from each channel within each pixel is then plotted in the graphs that you see here again from the tumoroid on day 0, and then here from the tumoroid on day 5. On day 0, we can see the extent of the signal in micrometers covered by the GFP expressing breast cancer cells and RFP expressing fibroblasts matches up precisely to each other, as well as with the total area of the tumoroid, as we can see with the bright field signal. However, after 5 days of incubation, when we perform the same type of analysis, it's clear that the breast cancer cells, which you can see with the green line, have migrated to a much greater extent than the fibroblasts. So again, it allows you to get a clear determination of how each co-cultured cell type is migrating in response to the chemoattractant or chemoattractant and inhibitor that's been added. Moving on from the initial invasion analysis, we also performed 2 different types of mechanism of action studies, as not only is it critical to examine the phenotypic invasive properties of cells and the interruption of that invasion, but it's also important to evaluate the different enzymes and binding activities that could potentially be involved in the invasion process. In the first that we're showing here involved monitoring the activity of matrix metalloproteinases, or MMPs, that have been shown to play a role in the tumor invasion process. This was done using a novel protease activity detection technology from Enzium called NSense. The assay technology based on a proprietary protein substrate, which contains a selective recognition sequence for a specific protease, in addition to a fluorogenic dye, works in the following manner. First, as we see in the first cartoon, the horseshoe-shaped substrate and dye are added to the well. Second, in the presence of the appropriate protease, the substrate is cleaved, exposing the dye binding site that you see here. And then upon binding of the dye, a far red-shifted signal is then created. Therefore, little to no fluorescence is seen when the substrate is not in the presence of the correct protease. The assay components were able to be incorporated into the existing 3D tumor invasion procedure in a simple, straightforward manner. The substrates and dye, as you can see here, were added to the diluted matrigel matrix containing chemoattractant, or again, chemoattractant and inhibitor. After addition to the aqueous layer and centrifugation, the components disseminate through the entire area surrounding the tumoroid. In this way, as the active proteases begin to cleave through the matrix, the substrate is also cleaved, which allows for binding of the dye and an increase in the red signal being emitted from those portions of the well. The first step in the project was to test the ability of the assay components to work properly within the context of the 3D tumor invasion model, as I just explained in the previous slide. The MMP14 substrate was selected for inclusion as this particular MMP has been shown in the literature to play a critical role in conferring cells with the ability to penetrate the extracellular matrix and is directly linked to tumorigenesis and metastasis. Again, here we see images that were captured over a multi-day incubation period, starting from day 0 through day 5. This is, again, specifically of the red signal emitted with the binding of the dye to the substrate. The Sci5 imaging cube was used to properly excite and capture the emitted signal from the dye. As this is a redshifted dye, and at times the signal from these dyes in its wavelength range can be somewhat weaker than dyes in the blue or green range, we chose to perform the imaging using 10x magnification instead of 4x. This allowed us to be able to capture the signal from the dye with greater sensitivity than would have otherwise been achieved. However, since the invading tumoroid structure covers a large area of the well, as you've seen in the previous slide, multiple images were needed to capture the entire structure. And we accomplished this by performing a montage imaging step and then automatically having the images stitched back together into the final pictures that you see here in the slide. And as you can see, as is expected, we start out with a minimal signal on day 0 immediately with the images that was captured immediately after the chemoattractant and inhibitor and the matrigel matrix. And then this signal dramatically increases over time until after a 6-day incubation, we see a very dramatic increase in the signal being emitted from a large area of the tumoroid structure. As matrix metalloproteinases have been shown to be active in the invadipodia portions of the tumoroid structure, specifically MMP14, we also compared the total coverage of the observed signal from the dye to that of the complete structure by overlaying the Sci5 and bright field images, which is what you're seeing in the image here. And again, as you can see from this image on the left, the extent of coverage observed with both Sci5 and with the bright field signal match up quite well, showing that the MMP14 proteinase is active in pretty much every area of the invading tumoroid structure. Then if we look closer at the images on the right, shown here, highlighted with the yellow areas, and then shown here, as well as multiple areas with this picture, we can also see that the signal increases in the invadipodia, meaning that MMP14 is indeed quite active in these leading portions of the invading tumoroid structure. We can also perform cellular analysis using the Sci5 signal from the dye, and as you can see here with both the images and the graphs that I'm showing on this slide, the object masks are drawn in a manner similar to what I explained previously. So again, you're not using GFP or bright field signal, but actually using specifically the Sci5 signal to have Gen5 draw the object masks around the parts of the tumoroid structure meeting the analysis criteria. Then using the results that were calculated, we can compare either the area coverage shown in the graph on the left or the total Sci5 signal seen here on the graph on the right within each object mask. This then allows you to specifically quantify the extent of MMP activity over time. The next step, once we confirmed that the assay was performing correctly and signal was indeed observed in the expected areas of the structure, was to add an MMP inhibitor to confirm that cleavage of the substrate was indeed from MMP activity and not from being in the presence of the components of the matrigel matrix. Here we used GM6001, which is a broad spectrum MMP inhibitor. The inhibitor was added as I previously explained, and as you can see from the images shown here, there is a concentration-dependent decrease in the signal from the MMP14 substrates and dye. The signal is never completely eliminated, however, as the original tumoroid is never completely destroyed, which is similar to what has been observed in vivo and is most likely due to the fact that the inhibitor does not completely penetrate the tumoroid. The extent of inhibition can then be quantified once again using the same type of analysis procedures that I've mentioned, and confirms again that interruption of invasion as well as an inhibition of MMP14 activity is indeed concentration dependent, as you can see here with a decrease both in the area of the tumoroid that you see the red signal, as well as an actual decrease in the signal captured with the Sci5 imaging cube with increasing concentrations of the GM6001 and shown with the decrease in the values in the two graphs. And this can accurately also be detected and measured through the incorporation of the Citation 5 and the Gen 5 data analysis software. The last step in the project was to confirm that we were seeing selective cleavage of the added substrate and subsequent signal generation from the target MMP. Here we incorporated selective substrates for MMP14 and MMP2, as well as the no-pro substrate, which is cleaved by a yeast protease and which is not expressed in mammalian cells, which obviously made it an ideal negative control substrate to use in this case. Two different types of inhibitors were tested. Again, GM6001, the broad range MMP inhibitor, and oridonin, which is a selective inhibitor for MMP2 and MMP9. And if we look at the results first for GM6001 on the left here with these graphs, we see that the compound inhibits substrate cleavage and subsequent signal production in a dose-dependent manner when both MMP2 or MMP14 substrates are incorporated. And you can see here with the decrease, again, in both in the Sci5 signal from the that was included in the masks that were drawn around the objects, both for MMP14 here and then MMP2. Then if we look at the results from oridonin, shown here on the right, again, the Sci5 signal is inhibited in a dose-dependent manner when the MMP2 substrate is added, shown here. Again, you can see the similar type of shape and descending values of Sci5 signal, similar to what we saw here with GM6001. However, when the MMP14 substrate was incorporated, shown here, no discernible inhibition is seen until the highest concentration was reached, which is approximately 30 micromolar. And by looking at other images that were captured from that well, the loss in signal was determined by us to be from a cytotoxic effect from the compound and not due to true inhibition, which agrees with published literature stating that oridonin exhibits an apoptotic effect on MDA MB231 breast cancer cells. And then finally, if we look at the results from the no-pro substrate with each compound shown by the two graphs here, we see that only low levels of signal are generated, which would indicate that the substrate is not being cleaved in any appreciable manner. Then a final confirmation that was performed to help ensure that the decrease in Sci5 signal was not just from the fact that the object mask was decreasing in size upon increasing compound concentration was to look at the total Psi5 signal generated within the entire image. So this is a different type of analysis that I've been showing to you, where we're not drawing any object masks around any portions of the tumoroid structure, but looking at the actual total Psi5 signal that would be emanating from the entire image. So closer to the type of analysis that you would use for a typical microplate read using a PMT type of technology. As I mentioned, this eliminates the use of the cellular mask and incorporates the signal from every pixel. And again, we see the same types of decrease in signal with both the MMP14 and MMP2 substrate, with increasing GM6001 concentrations, as you see here with the graph on the left. However, the dose-dependent decrease with the addition of orodonin is only seen in the results from the MMP2 substrate, shown in blue. And then again, there's really no appreciable change shown with the bars in red from the MMP14 substrate. And this again confirms that accurate results can be achieved when combining the NSense assay components with the 3D tumor invasion procedure. The second mechanism of action project was performed to study the correlation of ligand binding to the CXCR4 chemokine receptor, as well as interruption of that binding, and then how it relates to the final invasive phenotypic effect. The expression of CXCR4 is tightly correlated with the metastatic properties of breast cancer cells, and in vivo, neutralizing the interaction of CXCR4 with its known ligand SDF1 alpha, also known as CXCL12, significantly impaired the metastasis of breast cancer cells and cell migration. Traditionally, the discovery of novel agents has been guided by the affinity of the ligand for the receptor under equilibrium conditions, largely ignoring the kinetic aspects of the ligand-receptor interaction. However, awareness of the importance of binding kinetics has started to increase due to the accumulating evidence suggesting that the in vivo effectiveness of ligands may be attributed to the time a particular ligand resides at its receptor, or otherwise known as the drug target residence time. Due to these findings in this project, we examined the drug target residence time of various CXCR4 inhibitors in relation to the known ligand using the Teglite direct homogeneous receptor ligand binding assay that you see here from SysBio. And in general, the assay works in the following manner. Cells are engineered to express the receptor of interest, which you see here, which contains a snap tag labeled with a terbium molecule. A labeled ligand, shown here, is then added to the well, and as binding occurs, energy is transferred from the donor floor to the acceptor, and then this causes the ratio of the signal from the acceptor compared to the donor floor to then increase. The ratio continues to increase as the number of binding events also increase, eventually reaching a plateau over time, which again, as previously mentioned, is usually what has been monitored with these types of assays. Then if a molecule is introduced, such as shown here in green, introduced to the well, which interrupts the binding, this molecule preferentially binds to the receptor, kicks off the labeled ligand, and FRET does not occur, and then the ratio of the acceptor to the donor floor then decreases subsequently. The final residence time value takes into account the observed on and off rates of the unlabeled inhibitors, obviously, and is computed by the incorporation of the Matulsky and Mahan equation. The on and off rates of the labeled ligand are also necessary components of the equation. Therefore, an associative binding experiment was first performed using the acceptor floor labeled ligand that I just talked about previously. Then by plotting the HTRF ratios, those values over time for each of the ligand concentrations tested, which you see with all the different curves that have been plotted here, the observed binding values, or the K-obs, can then be generated. The KD value of the ligand can also be determined by plotting the HTRF ratios generating after a binding equilibrium was reached. Again, as I showed here, therefore the final HTRF ratios would be used from this portion of the curve and then plotted here to generate the KD value that is then shown. We were also able to confirm the law of mass action by plotting the calculated observed association binding values, or the K-obs values, that's shown here on the graph on the right in relation to ligand concentration, and observing a linear relationship between the two, as you can see here. The association and dissociation rate constants, or the K-on and the K-off, using the linear regression line that was calculated, can then be generated. The residence time of the unlabeled inhibitor panel was then determined in the subsequent experiment. Here, the labeled ligands and unlabeled competitors were added to the well at the same time, and then co-incubated with the CXCR4 expressing cells during kinetic association experiment. From the curve fitting and the Matulsky and Mann equation that I mentioned previously, the K-on and K-off kinetic constants of the unlabeled competitors were then generated. As you can see here, the residence time shown with the values that I've circled in red were then derived by dividing 1 by the K-off value. These values were then compared to the residence time of the labeled ligand, which you see here. From these values, as well as from the shape of the curves for the concentrations of inhibitor tested, qualitative assumptions regarding the constant can then be made. First, if the competitor dissociates faster than its target, from its target than the ligand, such as we see here in the results when testing AMD 3100 and WZ811, the specific binding of the ligand will slowly and monotonically approach its equilibrium time. And this then leads to a low resonance time, such as the values that are shown here. However, when the competitor dissociates slower, such as we see with AMD 3465 and IT1T, the association curve of the ligand will consist of 2 distinct phases, starting with a typical overshoot, such as what you see here and here, and then a decline until a new equilibrium is reached, such as we see here. Compounds whose resonance times are greater, again, which we see by the resonance times generated for these 2 compounds. Again, the values that are greater than that of the known ligand, which was 0.44 for the labeled SDF1 alpha, may then exhibit a stronger inhibitory response when used in the confirmatory phenotypic assay, which is then what we went on to perform. The inhibitor panel used in the resonance time study was then run with a 3D tumor invasion assay procedure, such as I previously described, in the presence of a single concentration of unlabeled SDF1 alpha. And here you can see an example of the co-cultured tumoroid structures, again on day 0. And then after a 5-day incubation in the presence of the chemoattractant and either AMD 3465, which is shown by the images on the top here, or in the presence of CTCE 9908, such as the image you see on the bottom, whose residence time was shorter than that of the ligand. And by the images that were captured of the tumoroid structures in the presence of AMD 3465, it's plain to see that the compound is exhibiting dose-dependent inhibition of final tumor invasion, whereas, again, CTCE 9908, whose resonance time value was shorter than that of the ligand, exhibits no discernible inhibitory effect on invasion at any of the concentrations that were tested. The effect of residence time was further confirmed by observing the results from all 6 of the compounds that were tested. IT1T, again, whose resonance time is longer than that of the ligand, similar to AMD 3465, also inhibits invasion in a dose-dependent manner, such as what you can see here, again, similar to AMD 3465. While all the other compounds that were tested exhibits really little or no ability to interrupt invasion through the 3D matrix, and that's shown by the results that you see with these 4 graphs here. So in conclusion, the slides and results that I've shown today hopefully confirm how proper instrumentation, microplates, cell models, assay chemistries, and testing procedures can provide combined processes that are both easy to perform provide accurate, reproducible data to better understand the target-based and phenotypic effects of potential inhibitors of tumor invasion and metastasis. If you'd like any additional information specific to all the different products that were mentioned here, you can find that information by going to one of the websites that's listed here specifically for each of the different companies. So it looks like we have a little bit of time to go and look at a few of the questions that have come in during the presentation. So I'll do that now before we finish up. The first one states, when the spheroid is first introduced into the well, do you centrifuge the plate, and at what time and duration did you take? So the answer is yes. So it's important to remember that for centrifuging the microplates, the centrifuge should be cooled down. If you've ever dealt or had to use Matrigel, obviously you'll know that it's extremely important to keep the matrix cool so that it does not begin to gel prior to when it's been properly positioned within the well. So the centrifuge was cooled down to 4 degrees Celsius prior to the centrifugation process, and then for centrifugation, I usually would spin the plate for just a matter of a minute or two, typically at about 200 times G. Again, in a swing bucket rotor type of a centrifuge. This obviously can be optimized depending on the type of centrifuge that you have in your lab, and also depending on the type of cell models that you're using. Okay, the second is, do you have a list of cell lines responsive to CXCL12 in this assay other than MDA MB231? I do not. What we did was this project was run as a example or a confirmatory type of a project to show that this type of assay could be performed in cooperation with or with the inclusion of the spheroid microplates, the citation imager, the software, and then the different types of assay kits that were included. But I'm sure that through different types of literature searches, that different types of cell models could then be identified that are truly responsive to the to CXCL12 as a chemoattractant. The next one is how many NSense substrates are available. That question is best answered by the experts at Enzium Labs. And again, you can go to their website, as I've shown here under the additional resource slide, and they have a nice list of the different substrates that are currently available to use with the assay. And then the next question is, how deep can the microscope image inside of the tumor spheroid? That's a good question, and as I mentioned previously, this is a wide field type of microscopy. So the actual amount of penetration of the light into the tumoroid structure is not going to be as complete as other types of microscopy. However, with this type of assay, again, we're looking at invasion that's moving out in all axes. So therefore, again, with this type of analysis, in my opinion, the actual penetration or the ability to actually see the inside, the true inside of the tumoroid structure is probably not going to be as critical as with other types of cell analysis that incorporate different 3D type of structures. So that, again, the type of analysis and the Z projection images that we were able to create using the Citation 5 and the Gen 5 analysis should indeed be able to give you, again, a nice accurate account of the invasion that's going on within the wells. And then let's see, I think that's about all the questions that I see at this time. If we do see others come in, we will certainly pass those on. Either I will answer them myself, or I will do my best to pass them on, again, to our friends at Corning, Enzium, and SysBio to make sure that the question is being answered in the best way possible. So with that, I will turn it back over to Lindsay.
Great. Well, thank you very much for joining us today, and thank you to Brad for his presentation and for taking time to answer some questions. We will email this recorded event to all participants. If you have further questions for our speaker or would like more information on BioTek's products, please visit our website. In addition, please check out our blog, like us on Facebook, and follow us on Twitter to hear more about BioTek webinars or to learn about our products and how they can help you. Again, thanks for joining us.